Welcome to this lecture on the PCR-Oligonucleotide Ligation Assay, a cornerstone technique in animal biotechnology. Imagine trying to find a single typo in a library filled with billions of books. That is exactly the challenge biologists face when looking for genetic mutations. Today, we are going to explore how this brilliant molecular tool allows us to pinpoint these microscopic errors with absolute certainty, helping us diagnose genetic diseases before they even show symptoms. Let us dive in. To understand how this technique works, let us start with a simple analogy. Imagine you are trying to repair a broken bridge by welding two massive steel beams together. The welder can only create a strong, lasting bond if the two edges of the beams align perfectly, down to the microscopic level. If one edge is even slightly jagged or mismatched, the welding torch will fail to join them, and the bridge remains broken. In the microscopic world of our cells, we use this exact same principle to find tiny hidden errors in our DNA. We use a biological welder to test if two pieces of DNA fit perfectly together. Now that we have our welding analogy in mind, let us look at the actual biological process. The PCR-Oligonucleotide Ligation Assay, or PCR-OLA, is a highly sensitive technique used to detect single nucleotide polymorphisms. These are tiny, single-letter changes in the DNA sequence. The process begins with the polymerase chain reaction, which acts as a biological photocopier to massively amplify the specific region of DNA we want to test. Once we have millions of copies, we introduce two short, custom-designed pieces of DNA called oligonucleotides, or probes. These probes are designed to bind side by side, directly over the exact spot where a suspected mutation might be located. With our probes in place, we now introduce our biological welder. An enzyme called DNA ligase is added to the mixture, which attempts to join, or ligate, these two probes together into one continuous strand. This is where the magic happens. The ligase enzyme is incredibly strict. It will only join the probes if they perfectly match the underlying target DNA without a single microscopic gap or mismatch at the junction. If the DNA is normal, they weld together perfectly. But if there is a mutation present, the edges will not align. The ligase will fail to join them, and we immediately know a mutation is present. You might be wondering why we go through all this trouble instead of just using standard DNA testing. The reality is that many devastating genetic diseases, such as sickle cell anemia and cystic fibrosis, are caused by a mutation in just one single base pair out of the 3 billion base pairs in the human genome. Standard PCR alone is fantastic at making copies of DNA, but it is not precise enough to easily distinguish a one-letter difference on its own. The PCR-OLA method provides the extreme pinpoint accuracy required to confidently diagnose these genetic conditions before severe symptoms even appear, making genetic counseling and early intervention possible. To truly master this technique, we must understand the mathematics behind it, starting with the amplification step. This formula models exponential growth during PCR. The variable n represents the final number of amplified DNA molecules available for the ligation assay. N0 stands for the initial number of target DNA molecules extracted from the patient. E represents the efficiency of the PCR amplification, which is a decimal between 0 and 1, indicating how well the DNA copies itself. Finally, N is the total number of heating and cooling cycles. If the efficiency increases, or if we run more cycles, the final amount of DNA grows exponentially, ensuring we have enough material for the ligase enzyme to work with. Let us put this amplification formula into practice with a concrete example. A laboratory is testing a patient, starting with an initial DNA template count of 500 molecules. The PCR machine runs for 25 cycles with an amplification efficiency of 0.85. We substitute these known values into our exponential growth equation. We add the efficiency of 0.85 to 1, giving us a growth factor of 1.85 per cycle. Raising this to the power of 25 and multiplying by the initial 500 molecules gives us approximately 2.37 billion total DNA molecules. This massive number shows exactly why PCR is a necessary first step to generate a detectable signal. Now that we have billions of DNA copies, let us look at how we measure the final result. This formula models the diagnostic signal we detect after the ligation step is complete. The variable S represents the total detectable signal, usually measured as fluorescence emitted by successfully joined probes. The constant K is a machine-specific calibration factor that converts ligated molecules into readable units. The variable M is the crucial match factor. It equals 1 if the patient's DNA perfectly matches the probes, and equals 0 if a mutation causes a mismatch. Because M acts as a multiplier, a mismatch forces the entire signal S to become 0, clearly indicating the presence of the mutation to the technician. Continuing with our healthy patient, let us calculate the final fluorescence signal. We know the patient has healthy DNA, meaning the probes match perfectly, so our match factor M is exactly 1. The laboratory equipment has a calibration constant K of 2 × 10 to the negative 7. We plug in this constant, our match factor of 1, and the 2.37 billion DNA molecules we calculated earlier. Multiplying these values together yields a final signal of 474 fluorescence units. Physically, this high positive number confirms that every single one of those billion DNA molecules successfully ligated, proving the patient's DNA sequence is perfectly normal at that specific location. We have seen what a perfectly healthy result looks like, but what happens if a patient is a carrier for a disease? A patient being tested for sickle cell anemia might be heterozygous, meaning they have one normal gene copy and one mutated gene copy. In this scenario, their amplified DNA sample is a perfect 50-50 mixture of normal and mutated sequences. Therefore, when we apply the normal diagnostic probe, it will only find a perfect match on exactly half of the amplified DNA molecules. To represent this biological reality mathematically, our effective match factor M becomes 0.5 because the ligase enzyme will physically reject half of the DNA templates. Let us calculate the expected signal for this heterozygous carrier. In this new test, the PCR step generated 400 million DNA molecules. We use our machine constant of 3 × 10 to the negative 6, and crucially, we substitute 0.5 for our match factor. We multiply the constant, the half match factor, and the total DNA molecules. The final calculated signal is 600 fluorescence units. Physically, this means the machine detects exactly half the signal it would expect from a completely healthy person, who would have generated 1,200 units. This intermediate signal perfectly informs the doctor that the patient is a carrier, possessing one healthy allele and one mutated allele. To solidify our understanding, let us look at a visual representation of the entire mechanism. In this diagram, you can see the long target DNA strand at the bottom, which we amplified using PCR. Bound directly to it are our two custom probes. The left probe features a capture tag, and the right probe features a reporter tag, which generates our fluorescence. Notice the critical junction between them, labeled as the SNP or mutation site. Hovering right above this junction is the DNA ligase enzyme. The diagram captures the exact moment the ligase checks the junction. If the match is perfect, it welds the capture and reporter tags together, allowing us to extract and measure the glowing reporter signal. We have covered a lot of ground today, so let us review the most important ideas. The PCR-Oligonucleotide Ligation Assay is a brilliant fusion of two powerful biological tools. By first amplifying the DNA with PCR, we ensure we have enough material to measure. Then, by relying on the extreme strictness of the DNA ligase enzyme, we can detect genetic mutations down to a single altered letter. We saw how the match factor mathematically dictates whether we see a full signal for a healthy patient, a half signal for a carrier, or zero signal for a mutation. This elegant combination of brute force amplification and pinpoint precision makes it an indispensable tool in modern molecular diagnostics.